Hello, my name is Joshua Gao, and today I will be doing my final for microbiology, which is a verbal exam, going over the questions that were given to me. For the first question, and for background, I have the questions behind me, but just to make sure I am asking the correct question at the correct time. The first question asks, what was my favorite lab, and or did I choose my favorite lab based off of my future career, or did I choose it due to pure enjoyment? And for this one, I would say both. It would have to be the hand washing lab. So I went to be a nurse when I grew up, so PPE, hand washing, and hand hygiene is very important to me. So it was really fun to experience that lab and then to also have others participate in it with my friends and roommates. So it allowed for less of a lab, but more as like a fun learning and true way of learning more about microbes, pathogens, and how all of those come together with microbiology. For the second question, it asks, if you had an unknown microbe, which steps would you take to determine what type of microbe, fungi, bacteria, or virus is it? Are there particular characteristics you would search for and explain? So the first thing that I would do if I was given an unidentified microbe, the first thing I would do is make sure I have all my PPE on and all my counter spaces is cleaned before I would work with it. And the first thing I would do is get the, microbe and then incubate it into some nutrient broth. And then once that has shown signs of growth, I would then use, over a couple day period of time, I would then use that culture that was utilized and then swab it on agar plates. Since I have leftover agar plates, I would probably do 3. In this case, I would do just 1 swab, just so I can see a base agar, and then I would swab the other 2. And if I had the other 2 agar plates, I would also use antibiotics to put them on the plate as well, like for the Kirby-Bauer method, to see what kind of antibiotics work against it, then to rule out, and then also have other factors such as temperature come in to see if incubation was at a high temperature and therefore growth was more, or if it was better growth under other or lower temperatures. So there's just many factors that I would take in, and also gram stain, so I could see what color it is. That would also have another big impact on what, or an identifier on what that would be. And yeah. As we move on to the third question, it asks, imagine you are interacting with an individual that does not think vaccines are effective. Using your knowledge of immunology, epidemiology, and microbes in general, describe how you might converse with them to educate them on vaccines. So first thing I would preface that whether you choose to get vaccinated or not is your decision, and at the end of the day, it will always be your decision. When people sometimes talk about whether they are or are not vaccinated, it can get kind of hostile. So it's always to make sure that it's an open-ended conversation, allowing no judgment, essentially. So you really just need to start off the whole conversation with open ears. And then once you get from there, you need to start asking questions of why you start to feel this way. Let them fill you in. Let them do the talking to understand their concerns of why it may not work and the reasons it just may not work out. And once you get from there, you share reliable sources of information that can prove the vaccine can be beneficial and helpful. And you can do so by helping them find websites that end in .org or .gov, because those are government regulated and far more trustworthy than the basic or average website. And then after that, you just need other reasons to get vaccinated. Friends, family, the safety of other people. Vaccines aren't meant to be scary. Vaccines are essentially just a tool to be used to help prolong your life and others around you. While the body does produce things that can help fight off vaccines, pathogens, and microbes, those aren't necessarily the best for first line of defense. It should be taken lightly that when you come into contact with a microbe, it should be taken seriously, and you should practice hand hygiene and other factors to eliminate this potential disease. And the reason why, and a vaccine is essentially just composed of, for example, the flu. The flu is a very common disease that we all get. It's nationwide throughout America and very prominent, so something we're all familiar with. The flu vaccine is essentially a lesser version of what the true form is. So it's a weak version that we pretty much inject into our body, so our body understands that there's a bacteria and the body needs to start producing white blood cells from our immune system to help fight off this. And with this surplus of already fighting off flu-like bacteria and strains, it will already be ready to fight off the flu if it comes in direct contact, which is hence the reason of vaccine shots, such as the booster for COVID and why it has been administered, and why you should probably get multiple shots. And you're allowed to get multiple shots unless you got the Johnson & Johnson, of course. But vaccines may seem scary, but the only thing scary about them is the lack of knowledge that you may have on them. It's once you do more reading on case studies and actual sources that are reliable, you can easily determine that vaccines are important and they can help you and save other lives around you. Hello. As we now get to question 4, it asks us to describe at least 2 positive and 2 negative relationships humans have with microbes, and then it asks to describe some of those advantages and disadvantages for the relationship. So as we begin to look at the positive aspect held between humans and microbes, we can see that bacteria sometimes help protect you when not all bacteria are bad. So there are bacteria that live inside your body and are totally helpful, and they will protect you, and even go as far as lining in your intestine and maybe even repairing tissue damage or other things that may cause you harm or hurt. And that's obviously a huge advantage, but the only disadvantage is to the microbe, and where they're not allowed to come into the host and then run its course, whether it is mild or deadly. As we get to the next positive relationship between microbes and humans, bacteria have been proven to be useful to break down sugars and carbohydrates and other toxins, allowing us to digest fatty acids, and which is a huge advantage, allowing us to digest and go through our whole, and then cleaning out our system essentially, and also allow it to burn sugar and other things and that allow us to move and function as a human. And now we can get to the negative relationships between humans and microbes. So while some bacterias may be harmful, there are microbes that are sought out to kill us, such as cancer, tuberculosis, diabetes, cardiovascular disease. There are numerous disadvantages set against us, and so one advantage to that is how since we've had such a large outbringing of those things, we've now had mass studies done that allow us to essentially help us overcome these obstacles and diseases and horrific pathogens, and allow for a healthier and prosper life. And as we get onto the next negative relationship between humans and microbes, although a bacteria hypothetically was to be in you, it doesn't need to have a large presence to necessarily have a large effect, meaning that maybe one microscopic worth of that bacteria can be enough to kill you. So the size amount of that disease does matter and has some factors, but once essentially it is in you, such as HIV or a disease that's in you, it stays with you forever and begins to reproduce, such as this cancer and other horrific pathogens, which are complete disadvantages at us. But one advantage that we have to that is sometimes with these slowly growing bacterias, they can also be eliminated early on, so we can eradicate them and then getting us back to a healthy and positive life. As we get to the last question, it asks, how many applications of microbes exist? Please list 5 uses of microbes. And so the first thing that I think of is the Kirby-Bauer method, and how we first got the microbe and incubated it and growed it through nutrient broth, and then we essentially, once it showed that it was a positive culture and there was a bacteria growing in it, we would then streak that onto a agar plate and then put discs on it to see whether antibiotics would react differently, show signs of growth, color, and then we would add things like temperature into account and other factors as such. Another way microbes can be used is through lactic acid, which we used recently in a lab where we helped create cheese, yogurt, and other basic foods that are kind of like dairy product or dairy based, essentially. Microbes are also everywhere. We are made of microbes. Half of our body is, and it makes up essentially half of us. Without them, we would be nothing and we wouldn't even be here. We wouldn't be able to digest properly or go through, life, essentially. Microbes are also in the air. They're in our oxygen and even in proteins. And finally, microbes can also be found in other foods, such as yeast, and to make bread, wine, and beer. Microbes are essentially just a fundamental part of life. We need them to survive, and they will always need us to survive. It's a codependent relationship that's been going on forever and allows essentially a harmonious but yet taxing relationship.